Thanks everyone or thanks for the organizers to invite me to talk in this workshop. I've actually attended the one in 2021 as well. So I suppose this is kind of an update since last time. So I'll give an overview of what we've been doing here at Cambridge University. There's obviously a lot of overlap in both the content and the challenges that we're facing. So I work at the bioinformatics training facility at Cambridge University. So we have a dedicated team here that does training in these sorts of courses that we're talking about, but also more foundational courses like Intro to Unix, Intro to R, Intro to Python, those sorts of things. So the single cell course specifically is in most of the work really has been done by the Cancer Research UK. Researchers there that work in, or not researchers, they're actually bioinformaticians in the core bioinformatics facility there. So most of the work really has been done by them. So the course that we run, just to give you an overview, and I'll start straight away with the disappointment that we are in R and we're using probably the least popular ecosystem, which is the bioconductor ecosystem. So it's a 3-day course at the moment that starts with the general introduction to single-cell RNA-seq technologies. And then we move on to the practicals that focus on 10x data. And like I said, we're using the bioconductor ecosystem of packages. The reason is very simple. Most of the trainers and the developers of the materials work with bioconductor. John Mariani is at CRUK. Aaron Loon used to be at CRUK, developed a lot of the packages early on. So this is what we're used to using. And that's what we've developed the materials around. The other reason is that, because I know people were discussing the languages, but one reason I think, or one advantage is that, for example, when you teach the bulk RNA-seq course, I think it would be difficult to teach that in Python. I know there's been a recent translation of the DE-seq package to Python, but a lot of the participants have already been exposed to some bioconductor in other courses. So it's kind of nice that this kind of follows on from those courses. There's also the OSCA book, the Orchestrating Single Cell Analysis book, which is a huge resource actually, and we kind of use that as a textbook almost during the class. The topics we cover, they're kind of illustrated here in this scheme. It's pretty much the standard topics you would expect, especially the early steps of the analysis are pretty standard. And then we talk about differential expression and differential abundance analysis. The data set that we use comes from this paper, so it's data from childhood leukemia. And the reason to pick this data is that you have diverse cell types in a well annotated system. So it's good when you're doing the clustering and marker genes that you can kind of pick on things from the literature and genes to look at. There's also samples from different patients, so you can see some batch effects there. So it's nice to illustrate the data integration steps. And also they have different conditions, different types of leukemia, for example, and some healthy patients as well. So we can do this differential analysis sessions as well at the end. So I'll move on to that's the quick overview of the workshop, and I'll move on to the rest of the slides is basically challenges. So the first challenge is demand. We just have so much demand for these workshops. So we've ran 5 workshops. The first one was back in 2021. When I participated in this trainers workshop, I think we just run our first iteration of the course. And now we've done 5 with 309 participants in total. Most of these have been online courses, so we can bump the numbers of participants quite a bit, assuming that we have enough trainers to support the session. But you can see here, this bar plot is just showing the percentage of people on the waiting list relative to the number of available places in each course. And this was the first iteration of the course back in 21. And it seemed like we were getting a handle of the waiting list, but in January this year, we had 105 people on the waiting list. So it's just like, this is a huge challenge. This outlier here in September last year was an in-person course. So in person, mostly we're catering for the local audience, right? But online, I've checked, and more than half the people on this waiting list are external to Cambridge. And we've had people in the workshops. We have some Australian colleagues here, and we've had Australian people, for example, attending our workshops at 2 in the morning, learning about whatever graph-based clustering or something like that. So very dedicated, but also means that we're catering for a very broad, like a wide audience, right? So that's the challenge, I think, demand. And the solution is easy, just run more courses, right? But the problem is you need trainers to run more courses. And the course is quite dense in materials. Like it's a lot of material for the trainers to prep. It's quite specialized. Not every trainer is comfortable teaching this sort of stuff. So that's a big challenge, I think, that we have at the moment. The other thing is what Paulo also mentioned, the prerequisites and the amount of code involved in these courses. So we're dealing with complex objects, no matter what ecosystem you're on. They're pretty much, the data structure is pretty much the same, and it's not just a data frame. It's quite a lot more to get your head around. And we're often using like custom functions. We're iterating through lists, we're data wrangling, and so on. And so something like our Intro to R course, for example, which is a data carpentry style course, if you're familiar with those, it doesn't really provide enough prerequisites for when people then jump into a course like this. Even like, this little chunk of code here, it doesn't seem like there's a lot happening, but if you just came from the data carpentry course, there's a lot of stuff to unpack here. And so this was a lesson we learned early on in our first iteration of the course. We thought we could do like these kind of live coded type of courses, like the data carpentry and software carpentry courses, but we moved away from that because there's just not enough time to live code things in these workshops. So what we do now is a bit, I think, similar to what Paulo was just describing. We have scripts with the code already there. The trainer is going through the code, explaining what's going on in the different steps of the code. And then there's exercises when they can do some coding themselves. But the self-coding is much more limited than in the original iteration of the course. I think one thing to keep in mind with the coding aspect is that that's probably not where we're adding the most value, because there's so many really good online tutorials and vignettes, and like the OSCA book, for example, that we recommend to our participants. I think really the value is coming from explaining the concepts and the pitfalls of these analyses. So, for example, we're talking about t-SNE and UMAP, and like emphasize multiple times in the workshop that you use this for visualization, not to input in your downstream analysis. Or there's always this question in clustering. People are always asking, what's the correct clustering? And we have a little discussion around, there's probably no correct clustering. And it's about thinking about what we're trying to do with our clustering, for example. So I think that's really something to keep in mind, that even though there's a lot of coding involved in the workshop, probably a lot of the value is coming from the concepts and the things you should and shouldn't do. So that's one challenge. Another one is, of course, the software environment and the size of the data. So there's a lot of packages that we use in the course. This is a list of the packages we directly load. Of course, the dependencies is like huge. Just to give you an idea here, if you go to the data carpentry course, you basically get exposed to the tidyverse package. It's like now you come to this course and there's like a whole suite of packages that may sometimes have their own like little syntax quirks and so on. So again, this prerequisite thing is an issue, I think. And in terms of data, we've had to do a lot of work. I don't know how you guys in other courses manage this, but we had to do a lot of work in downsampling some bits of data, pre-processing a lot of the data, and to make sure that people can move from one section of the materials to the next, we ended up creating lots of R objects so that each section you can always load a pre-processed R object in case you didn't manage to run all the steps in the previous session. And it also allows us to, for example, like in some of the sections, we subsample our data to 500 cells so that we're not waiting 10 minutes for some clustering algorithm to run. But then we bump the number of cells for the differential abundance session, for example. So we kind of can control a little bit how many subsets of the data are moving across the course. But this is actually in itself a big challenge, like just managing all the different objects in the course and which objects are used in the different sections. Our training environment, we're very lucky at the facility because we've got a full-time person that takes care of all the computing infrastructure. We typically deliver these in browser-based environments. So all the data and the software is already set up on the machines. So for the early sessions, we're using Cell Ranger. We just have an Ubuntu, a very lightweight Ubuntu distribution that's served on the browser with Guacamole, and then an RStudio server that participants log in with the username and password. So that's kind of how we solve this issue of the software environment. Another challenge is the material updates. Like it's a lot of work to keep a course like this up to date, as I'm sure a lot of you will appreciate. You can clearly see this is our GitHub pulse. You can clearly see when a workshop runs and there's like a rush to do the update to the materials. And most of the work really is like streamlining the code, the exercises, improving the flow of the course. That's been most of the work that the developers have been doing on this course. And then occasionally there's like, additions either in like managing the object creation for the workshop or adding new sections, like recently Abby added graph-based abundance analysis for the differential abundance section using Milo. And recently Adam added some CRAT materials as well to kind of have this parallel, if you prefer to use CRAT, here's the translation. And the other thing is, even though the field is stabilizing a bit, there's still quite a lot of changes sometimes. So every time there's a new package update, then we need to update the material. So here's an example when I guess Aaron they had a new function to identify marker genes, updated their OSCE book, and then a year later, so it's a bit of a delay, but a year later, we have to update our materials to reflect the new functions and documentation. So this adds quite a bit of extra work. So we're not adding new material, but we're having to keep up with the current state of the packages. And then, of course, there's so much more to cover. And in our feedback surveys, people are always asking for more things. So, for example, we don't cover trajectory or velocity analysis at the moment. The convolution of bulk samples is requested quite often. Multimodal data is another. And of course, spatial omics, which is a whole new course. And we don't have anything local at the moment running. And keeping in mind that I suppose it's a time to develop and maintain these materials, but also they would add, each of these sessions would add another half day or something like that to a course that's already quite intense and quite a bit of time for participants to dedicate. I should say that the work that I'm showing this slide because I really appreciate all the work that the training developers do in this course. I've only contributed very little by comparison. And it's been really nice to see that from the first iteration in November to the last one this year, that the feedback is getting better. So I think like our course is getting a little bit more fluid with less hiccups and more streamlined. There's also the challenge of convincing people to fill in feedback surveys. We always get very few answers. I'm sure this is a universal problem. So that's all I wanted to say in terms of the overview of what we're doing here. So thanks for listening, and of course, lots of time for discussions, and a lot of thanks as well for all the trainers that have been involved. So these are local trainers. The ones in bold here have contributed a substantial amount of materials, and then in my team as well. In particular, thanking Paul, who takes care of all the computational infrastructure, and Matt is my manager. And there's a few links here if you want to check the courses that we run at the moment and so on.